Hi, welcome to Science Hangout from Science Mission. I'm Sadashiva Pai. We have today Dr. Mark Huising, Assistant Professor in the Department of Neurobiology, Physiology and Behavior, and Physiology and Membrane Biology with us. Uh, he's from University of California at Davis. Dr. Huising has won several awards, including Dutch Zoology Award in 2006 from Royal Dutch Zoological Society, Endocrine Scholars Award in 2007 from the Endocrine Society, Novo Nordisk Award for Endocrinology in 2011, Career Development Award in 2013 from Juvenile Diabetes Research Foundation, Helmsley Award in 2015 from the Endocrine Society and the Helmsley Charitable Trust, Individual Biomedical Research Award in 2015, and he was nominated for the Helmholtz Young Investigator Diabetes Award in 2016. In addition, Dr. Huising is a member of several societies and has published several articles in well-known journals. Welcome, Dr. Huising.
Thank you.
Thank you for joining the Science Hangout.
Thank you for inviting me. It's a pleasure.
Let's talk about a little bit about your background, research interests, education at all, and all those things.
Sure. So I currently work at UC Davis, as you said. I did all my formal education in the Netherlands, both my Masters of Science in Biology and my PhD. I did my PhD at the University of Nijmegen on a different topic than what I'm working on today. I used to work on the evolution of immune and endocrine systems. I've been working on soluble signals, hormones, cytokines, pretty much for my entire career. So that's kind of the red thread that ties it all together. During my undergraduate career, I spent some time in the US, in Portland, and really liked that experience and got an opportunity to go back to the US after my PhD. I was invited by Wiley Vale to join his lab at the Salk Institute. So, and when you get an invitation to go work and live in San Diego, it's hard to say no. So we jumped on the plane and went for a postdoc at the Salk Institute and ended up staying there for almost 10 years. And then from the Salk, I moved on to my current position at UC Davis.
Okay. So can you talk us a little bit about your mentors, role models in your research career?
Sure. So I had two. I had co-mentors for my PhD, Lidie Verburg van Kemenade and Gert Flick, and at two different institutions. I did my formal defense at Nijmegen, but I also worked at Wageningen University, which is a small but very good school with an excellent program in biology. And both Lidie and Gert really let me do my own thing, chart my own course, and that's something that worked out really well for me. So they gave me the independence that personally for me works really well. And they also were very supportive of me subsequently going to the Salk Institute. And Wiley, I mean, I can't say enough great things about Wiley. Wiley, like Lidie and like Gert, Wiley was a phenomenal mentor. Very hands-off. He really enabled you to take projects and design them and run with them, and always there in the background, always available when you had questions or needed some guidance. But other than that, it was yours to take and run and do good things with. And that was a wonderful environment. So I really learned a tremendous amount from Wiley. So yeah, those are the folks who coached me and who I look up to. And who I try to emulate in how I lead my group today.
Okay, and I know you are busy with your research and all, but just tell us about your other hobbies, if at all you do anything else other than the research.
Sure. I try to make time to actually get out of the lab. I mean, we used to be in SoCal, now we're in NorCal. Both are wonderful for the outdoors, so we like to hike, go on walks. Try to do some trail running every once in a while. I used to play volleyball. There's a little bit less beach volleyball in Davis than there used to be in San Diego. So, but yeah, we try and spend time outdoors as much as we can.
Okay. And Tell us about your lab people, grad student, postdocs.
Sure. So I have 4 grad students currently and a postdoc, and a staff scientist and a technician. And that's a good size group. The lab right now is, I guess, a little heavy on grad students. Some US labs are heavier on postdocs. The environment that I grew up in scientifically back in the Netherlands, the labs there are very much run by grad students, so it's an environment that I'm familiar with. And it's working out well, and we have a large number of undergraduates. We have a fantastic pool of undergraduates at UC Davis, and we have a number of them participate in the research, help out, and that's actually tremendous fun, because you get to teach them really early on in their career and inspire them, hopefully, to go out and at least appreciate the importance of what we do.
Okay. And how do you motivate them? I know, like, they need to have self-motivation, but sometimes when the experiments don't work, they really feel sad and all those things. How do you pull them back?
Sure. What I try and do for each one of them is to give them projects that I think are exciting, get them started on something that already is up and running to the point that it will lead to something, that it will lead to a successful project with a reasonable amount of certainty. And I usually advise them to not put all their eggs in one basket. So have at least 2 projects. One of them can be riskier, but if one of them doesn't work out or there is some setbacks, you can always turn to and get your motivation from the other project. And I think so far, I mean, you can ask me, but I think so far this has worked out well.
Okay. And how often do you interact? Like, is there any policy? Like, you have open door policy, or how do you work with them?
Yeah, I always tell them you can email, or you can come in every time. You can email whenever you have questions. I'll let you know if there is if the moment is not appropriate, or if I'm really busy, but that's rare. So I probably talk to each one of them on a daily basis, and we have a more formal meeting once a week.
And so you are interested in soluble factors and all those things. So how you really focused on diabetes eventually? What made you just to go in that direction?
That's kind of a coincidence. So as I was starting my postdoc in Wiley's lab, so Wiley Vale discovered corticotropin releasing factor, which is the main hypothalamic hormone that kickstarts the endocrine stress response, at least the hypothalamus pituitary adrenal axis. And he not only discovered CRH or CRF, he also discovered 3 additional peptides, the receptors for all of those. So he really charted that whole family over the course of a 30-year span. And by the time I joined his lab, they had identified all 4 of those peptides. One of them being a peptide that I still study heavily, which is urocortin 3. And it just so happened that urocortin 3 is abundantly expressed in beta cells in the pancreas. So we really kind of followed the peptides and try and decipher the biology that is controlled by the peptides. So that's how we got our start in diabetes research. And I'm forever grateful to the Juvenile Diabetes Research Foundation, because they were the ones who recognized this early on and were able to support us to the point that we could embark on the line of research that I still run today.
Yeah. So talk us more about your urocortin 3 work from your presentation, please.
Oh, okay. So let's see. Here we go. All right. So I decided to share 2 stories today that both have to do with urocortin 3, and they both have to do with local crosstalk and how local crosstalk controls beta cell fate and function. I'm just going to talk you through both of these stories. So just one slide background on the why we are interested in pancreas and in diabetes. We have, as most of you will know, 2 major forms of diabetes, type 1 and type 2, which are really quite distinct diseases. Type 1 is the autoimmune disease that destroys insulin-producing beta cells in the pancreas. Almost 200,000 Americans under the age of 20 have type 1, and by the time that this year is over, nearly 16,000 children will have been newly diagnosed in the US alone. And that prevalence of type 1 is increasing for reasons that we don't entirely understand. Average age of diagnosis is between 10 and 12 years of age. And that's a little bit different for type 2 diabetes. This is the disease often associated with lifestyle choices, but with a distinct genetic component to it as well, where patients have a relative insulin deficiency that is precipitated by peripheral insulin resistance. Beta cells try and compensate, are able to do so for some time, and at some point that compensation fails and diabetes ensues. The latest estimates from the CDC are that over 29 million Americans have type 2 diabetes, although not all of them are aware that they have this disease. And over 1 in 3 Americans has prediabetes, and this is a major healthcare crisis in the making, because there's a good chance that those 1 in 3 Americans will develop type 2 diabetes in the near future. And some of the common complications that are complications for both uncontrolled type 1 and type 2 are macro and microvascular implications, increased chance of heart attack and stroke, as well as microvascular implications which precipitate in kidney disease, the need to amputate lower limbs, chances to go blind, cognitive impairment, and together these really still cause a reduced quality of life and reduced life expectancy. So most people who study diabetes and who study the pancreas are interested in beta cells, because beta cells are the only source of insulin in our body. And whenever beta cells secrete insulin, this leads to reduction in blood sugar. I'm also interested in what else happens in the islets. The islets do not consist only of beta cells. There are also alpha cells, which make another hormone that's called glucagon. And glucagon is the opposite, has the opposite role of insulin, where insulin tells your body to store nutrients away for later use. Glucagon talks to the liver and causes an increase in blood sugar to prevent dangerously low hypoglycemia. Then there's a third cell type, and that third cell type is a cell type that I've been studying quite heavily because this is where the biology has led me. And that cell is called the delta cell, and the delta cell makes somatostatin. And somatostatin, wherever you'll find it in the body, is an excellent brake. It inhibits its target cells. In this case, it inhibits both insulin release from beta cells and glucagon secretion from alpha cells. And we had known this since the 70s, pretty much since somatostatin was discovered, but it had never been fully appreciated how delta cells are activated. And what we discovered is that a beta cell peptide, urocortin 3, is co-released with insulin. And every time beta cells secrete insulin to act on peripheral insulin-sensitive tissues, they also secrete some urocortin 3, which then activates delta cells, and delta cells then respond with an increase in somatostatin to bring the whole thing back down. So the research interests of my lab are this crosstalk, but also the heterogeneity of beta cells. Not all beta cells are equal. What does that mean? And this story has led us to appreciate that there is really quite a bit of plasticity in the cell identity of islet cells. And I'll talk about this in the second half of my hangout today. So the two stories I will share with you is how, on the one hand, urocortin 3 is an islet hormone that promotes negative feedback from delta cells. Within the islet, it responds primarily to urocortin 3 coming from beta cells, but it also is responding to other hormones, for example, ghrelin. Ghrelin had long been known to inhibit insulin secretion, but the mechanism had not been figured out. It turns out that the way that ghrelin is able to inhibit insulin secretion is by activating delta cells. Delta cells go on to inhibit the beta cell and curb insulin secretion. And then the second half of what we study has to do with the fact that urocortin 3, in addition to controlling local feedback, is an excellent maturation marker for beta cells. It turns out that beta cells, once they express urocortin 3, are really functional beta cells. So urocortin 3 happens to be a wonderful proxy to determine whether beta cells are functionally mature. And I'll talk about this at some length as well. So when we started looking at urocortin 3 and to try and appreciate its function, we've done a couple of exercises. In this case, what I'm showing you is a dot plot of basically all messages, all hormones and cytokines encoded by genes in the mouse islet. And I've ranked them by abundance on a log scale, and I've also made each dot proportional to its abundance. So of course, if you do this for the entire islet, insulin, and mice happen to have two insulin genes. Insulin is the most abundant soluble factor, followed by amylin and glucagon. And really, I think in the sense that glucagon and amylin and insulin are the only ones released in the circulation to act at distal sites. This is really where we draw the line, and what are classical hormones, and which would be just these three, and which ones are local factors that in some paracrine mechanism modulate the activity of beta cells and alpha cells. And this local role is true for both somatostatin and urocortin 3. But if you think about it, looking at this graph, after insulin and amylin, urocortin 3 is really the third most abundant signal coming from the beta cell, and we had no idea what it was doing. You can do this by gene expression. Here I'm showing you that you can also grind islets up and look at peptide content and arrive at a very similar relative abundance of the the four major hormones that I'll be talking about. And this is a quick plot showing that, as we all know, as soon as you promote insulin secretion by going from low to high glucose, you also co-stimulate the release of urocortin 3. And if you look at how urocortin 3 and insulin are distributed in the beta cell, this is some super resolution macroscopy, you can actually see quite nicely that both insulin in red and urocortin 3 in green co-localize nicely in yellow in the secretory granules of beta cells. So we knew pretty well where the signal came from, but it was really a struggle to find out what the target was for urocortin 3. We know it activates a class B GPCR called CRHR2. We did not know what cell type is the main site of the expression of CRHR2, and there is really no reliable antibodies for them. So it took us about 6 years to get an answer to this question. It required me making a beta cell reporter line that labels all beta cells in red, and crossing this to either an SST-CRE or a glucagon-CRE to lineage label all delta or alpha cells. And once I had these mice in place, I could fax purify beta cells in either delta or alpha cells. And with this in hand, we now have really nice transcriptomes for each of these cell types. So now we can really ask the question for any gene that we're interested in, what cell type or cell types are the sites of expression. And if we do this for a panel of GPCRs, we discovered that CRHR2, the receptor for urocortin 3, is selectively expressed by delta cells. And by the way, these data are publicly available. We host them in this format on our website, so you can make these graphs, these browser plots for yourself. All you have to do is be able to type in your keyword. Right. So another line of evidence that we had that pointed us to delta cells is when we looked at urocortin 3 knockout islets. These mice are really quite normal. They have only a fairly mild phenotype, but among the genes that are selectively repressed in urocortin 3 knockout islets are somatostatin, HX, which is a transcription factor that Klaus Kestner showed to be required for delta cell lineage formation, and retinal binding protein 4, which also is expressed by delta cells. Also, when we looked at delta cell content, SST content is down in urocortin 3 no islets, but insulin and glucagon content is not. So all indications are that these urocortin 3 knockout islets have a subtle defect only in delta cells. So we looked at somatostatin secretion and insulin secretion. When we look at insulin secretion, and when we look at urocortin 3 no islets, when we go from low glucose to high glucose, we see a very characteristic spike, which is called the first phase in insulin secretion, which is then followed by a sustained second phase. And as is evident from this figure, the first and second phase insulin secretion in urocortin 3 knockout mice is exaggerated. Now, as soon as we restore urocortin 3 by adding exogenous synthetic urocortin 3 peptide, we can rescue this hypersecretion of insulin. So what's happening here is we're reapplying SST secretion, we're reactivating SST secretion, and this then inhibits insulin secretion, brings it back down to normal. And we were very excited when we found this because a couple of years prior, it had been published that the somatostatin knockout mice have a very similar phenotype. In the absence of somatostatin, insulin secretion from SST null islets is excessive, is exaggerated, both in first and second phase. And as soon as you restore synthetic SST peptide, you can rescue this phenotype as well. So the fact that the loss of urocortin 3 phenocopies the loss of SST so nicely places them in the same feedback loop. So this is all knockout mouse. This is all based on knockout islets. So what about healthy wild-type islets? So this is an experiment where we investigated just that. This is simply looking at somatostatin secretion from healthy mouse, wild-type mouse islets. And I'll talk you through this figure. Basically, as we go from low glucose to high glucose, we see a significant increase in SST secretion. This has been known. When we add additional urocortin-3, we can potentiate this secretion. When we come back with an antagonist to urocortin-3, which is a peptide called A-stressin-2B that blocks but doesn't activate the receptor, we can block the effect of urocortin-3. But to our excitement, what we also discovered as we applied A-stressin-2B by itself is we can fully block the glucose-stimulated SST secretion. So this demonstrated that not just urocortin-3 can activate delta cells. This shows that endogenous urocortin-3 is required for the glucose-stimulated secretion of somatostatin. We can do the same thing in perfusion. Here I'm looking first at somatostatin secretion. As you go from basal glucose to high glucose, you see a marked increase in SST secretion. And as soon as we add A-stressin-2B, thereby blocking endogenous urocortin-3, you start to inhibit SST secretion. And consequently, as you look at insulin secretion, as we're starting to inhibit the negative feedback, we allow the de-repression of insulin secretion. So by inhibiting the feedback that is mediated by urocortin-3, we can actually cause the beta cells to secrete more insulin. These data show that in human islets, the same thing happens. We see both on the basal and high glucose that urocortin-3 can stimulate human somatostatin secretion, and A-stressin-2B is able to significantly block this effect. So this is not an effect that is unique to mouse. It also happens in human islets. So mechanistically then, where does that leave us? So if we first review how beta cells respond to glucose, beta cells in green, they take up glucose through glucose transporters in mouses, GLUT2. This glucose gets metabolized. Glycolysis, TCA cycle, oxidative phosphorylation leads to an increase in ATP. ATP then acts at K-ATP channels, closes them, and triggers depolarization, which is propagated then and leads to the opening of L-type calcium channels. Calcium flows in and triggers exocytosis of insulin, and this can be potentiated through hormones such as GLP-1, glucagon-like peptide 1, which mediates an increase in cyclic AMP through a class B G protein coupled receptor. This is how common drugs in diabetes like Byetta work. Now, what we think is that delta cells mechanistically work very similar in response to urocortin. So as we review what happens as we intervene in these key points in this pathway, as we go from low to high glucose, we see a very nice secretion of insulin. And as we either force the K-ATP channels in the open conformation, preventing their closure, we can fully block insulin secretion. As we block L-type calcium channels, we can do the same thing. So this demonstrates, as has been well known, that both K-ATP channels and L-type calcium channels are required for insulin secretion. Now, when we apply these same drugs and look at SST secretion, we see that both of these drugs also are able to inhibit glucose-stimulated SST secretion. But what we did not know at the time is whether this is because of a direct effect on delta cells, or whether this repression was mediated by the inhibition of urocortin-3 release from beta cells. So we then went back and applied synthetic urocortin-3. And if you apply synthetic urocortin-3, you can potentiate, you can stimulate SST secretion, which then predictably leads to a repression of insulin secretion. So now the question was, well, what happens if you apply diazoxide and isradipine in the presence of that urocortin-3? Can urocortin-3 overcome the inhibition by diazoxide and isradipine? And the answer is no. So this means that you need both K-ATP channels and L-type calcium channels at the level of the delta cell, in addition to the potentiating signal from urocortin-3. So what this then showed us is that mechanistically, the delta cell and the beta cell respond very similarly. They both respond to glucose and the ATP that is generated by glucose metabolism, and they both require K-ATP channels and L-type calcium channels to allow the exocytosis of their payload, SST in the case of delta cells, insulin in the case of beta cells, and in both cell types, there is the possibility to potentiate secretion via a peptide GPCR-mediated amplification path. All right. So we wanted to take this in vivo and intervene in endogenous urocortin-3 signaling. So in order to do this, we generated a reporter or a mouse that allowed us to overexpress urocortin-3, specifically in beta cells. So this is classic doxycycline-inducible mouse model, where we have a beta cell-specific driver of reverse tetracycline transactivated protein, which then can activate the expression of a urocortin-3 transgene under control of a doxycycline activatable promoter. So in short, you need to meet 3 conditions for a mouse to start to overexpress urocortin-3, specifically in beta cells. You need to carry both of these transgenes, and you need to administer doxycycline. So when we've done that, so these are the islets from 8 individual mice, and only the mouse that has both of the required transgenes and has received doxycycline will respond with a nice increase in transgenic urocortin-3 expression. And this is important. This transgenic urocortin-3 expression is at the same order of magnitude as the endogenous levels of urocortin-3. So we're not overexpressing by a wild margin. We're really hitting an appropriate dose that is on par with the endogenous levels of urocortin-3. And we're effectively doubling total urocortin-3 levels in this mouse. So with this mouse in hand, we went back and asked some questions in biology. And the first question that we asked is what happens if you start to express urocortin-3 earlier than normal? And something that I'll talk about in a little bit more detail later on is we had known that urocortin-3 at postnatal day 2, so after birth, isn't really yet expressed in most beta cells. It takes until about 3 weeks after birth before all beta cells, or at least the large majority of them, co-express urocortin-3 with insulin. And we had published this. And then Barak Bloom, when he was still with Doc Melton's lab, made a very interesting series of observations. He showed that this onset of urocortin-3 expression correlates with some really interesting changes in the beta cell threshold for glucose. So if you look at P15 islets, which are equivalent in their response to how adult islets will respond, if you take these islets and you take their glucose levels from 0.5 millimolar, which is hypoglycemic, to 2.8 millimolar, you typically would see no response. And as you take them from 2.8 or 0.5 back to 16.8, you get this very nice first phase and second phase secretion that I've also shown you. Now, if you do the exact same trace on postnatal 1, on islets from postnatal 1 pups, you see a very different pattern. Now, if you go from 0.5 to 2.8, you already see a first phase and second phase. And again, you see a similar response at 16.7 millimolar glucose. So these islets, these beta cells, are more sensitive to glucose. And this then translates into a higher level of insulin and consequently a lower level of blood glucose. And this type of behavior had been known for some time. What Barak was able to do is he was able to correlate this phenomenon with the onset of urocortin-3. Now, we've taken this one step further based on these observations, and we used these doxycycline-inducible mice to then prematurely induce urocortin-3. So we gave urocortin-3 to pregnant dams that themselves were incapable of overexpressing urocortin-3. And we basically were able to, only in bitransgenic pups, greatly overexpress the levels of urocortin-3 at postnatal day 2. And when we look at the plasma glucose levels of these mice, and this was done blind because we had no idea at the time we did this experiment which of the pups were the bitransgenic ones and which ones were the controls. But when we do this and we parse them out into either bitransgenic and DOX-induced mice, we can very nicely see that the wild type or the control mice took about 3 weeks of age to mature their blood glucose levels to the normal levels that we see in mice. But these bitransgenic DOX-induced mice, the ones where we prematurely induce urocortin-3, their plasma glucose values mature much sooner. And what's happening here is we are prematurely activating delta cell dependent feedback. We're prematurely inhibiting insulin secretion. And this prematurely allows plasma glucose to rise to levels that we typically see only in young adult or adult mice. As soon as endogenous urocortin-3 comes on, overexpressing a little bit more doesn't really make a difference anymore. Okay, so that's summarized here what we normally see in the early postnatal time period, where we have no urocortin-3. Plasma glucose is basically determined by the balance of insulin action and glucagon action. And as soon as urocortin-3 comes on, this activates negative feedback, and this antagonizes insulin action. So this then is responsible for the increase in plasma glucose levels to the levels that we typically associate with non-fasting plasma glucose levels in mouse. All right, so we then went to a disease model. We looked at OB mice. And OB mice, which are mice that are leptin deficient, they are morbidly obese, and they develop severe diabetes. They also lose the expression of urocortin 3 early on. We knew this. So what we did, and this then translates into a lack of SST secretion. There is really hardly any SST coming from these islets under any circumstance, meaning that the negative feedback control in these islets is pretty much all but gone. And we'll talk about what that might mean. But what we did is we took our doxycycline inducible mouse model, we bred it onto the OB background, and we followed these mice for about 6.5 weeks. At 6.5 weeks, we know that urocortin 3 levels are mostly gone. So at this point in time, we placed them on dox feed, and so we restored the expression of urocortin 3. And from the moment we did this, we saw this marked increase in plasma glucose levels, which is independent of body weight, which is unaffected by any of this intervention. So what's happening here, again, is we restore urocortin 3, we restore delta cell-dependent inhibitory feedback on beta cells, we probably inhibit insulin secretion, which then causes plasma glucose levels to actually go up a little bit. So by restoring this feedback, we actually exacerbate diabetes. So why have this feedback mechanism? Well, this is something that we wondered, and the way we think about this is that this is a nice way to restore or to counteract hyperglycemia and diabetes by shutting down negative feedback. But normally, like many things in biology, you need proper negative feedback control to have tight control over your parameter, and glucose is obviously no difference. So in order to visualize that, what we did is we outfitted mice with continuous glucose monitors. And the mouse that you see walking around here carries one of them on his back. This is a continuous glucose monitor that is used by people with type 1 diabetes. It sends a signal every 5 minutes to this handheld receiver, which normally isn't placed in the home cage, but we did so just for the purpose of this video. And this allows us to monitor glucose in free moving animals over time. And when we did this to both to lean mice over the course of 72 hours, we can see this very nice and tight control of a blood glucose. There's small incursions, but this is testament to how well a healthy pancreas works. Now, when we did the same thing to OB, morbidly obese litter mates, we saw that the average glucose were much higher. This is no surprise, we knew this. But what was surprising to us is the degree of volatility in these glucose profiles of OB mice. And to us, what this means is you no longer have the ability to timely attenuate insulin secretion, leading to an overshoot of insulin, which then needs to be compensated. So by losing that local feedback, you lose tight control over when to activate, but also when to shut down the beta cell and its insulin secretion. So I think in a healthy individual, this local feedback is really important to ensure that you have tight control over insulin secretion and therefore over blood glucose. All right, so does any of this hold in humans? And the answer is yes. So we looked at a cohort of human pancreas donors that we obtained from the ANPOD consortium. And in humans, there is a little species difference where urocortin 3 is expressed also by alpha cells. So what we've done is we stained human donors, we applied masks to define beta and alpha cells, and then we were able to isolate the intensity of the urocortin 3 stain in beta cells and in alpha cells. And what you can see, hopefully, in this particular set of examples is that in a human type 2 diabetes donor, beta cells are reduced, but the urocortin 3 intensity in these beta cells is also reduced compared to the situation in a healthy donor, whereas the intensity in alpha cells is far less affected. Now, human patients being human patients and being individual and therefore suffering from variation, we also were fortunate enough to collaborate with Lily Pound and Kevin Grove, and Kevin was still at OHSU, and they had this wonderful cohort of macaques that they had on either low-fat diet or a high-fat diet for a large number of for a number of years, and they shared these samples with us, and we did the exact same thing there. We stained their macaque islets for urocortin 3. We parsed out the intensity of urocortin 3 in beta cells and alpha cells, and we compared them between a control cohort on low-fat diet and high-fat diet fed macaques that either turn out to be sensitive to insulin, so resistant to the diet, or sensitive to the diet and therefore develop insulin resistance. So these are pre-diabetic macaques. They haven't progressed to frank diabetes, and already we start to see urocortin-3 a signal tapered off specifically in beta cells and be unaffected in alpha cells. So urocortin-3 is lost really early on, probably in pre-diabetes, both in humans and particularly also in macaques. So this feedback control that we discovered is not only relevant for rodent models, it also appears to be well in human disease. So to summarize this part, the point I want to make here, and that has been ignored, I think, a little bit, is that delta cells are really important. So I often get asked, well, are delta cells, there's only so few of them, how can they ever be meaningfully contributing to how insulin and glucagon are secreted? And I show them a video like this where you see all beta cells in red and delta cells, really a large number of them kind of enveloping that whole core of beta cells. And I think there's ample evidence to suggest that there is enough delta cells, and all they have to do is secrete enough somatostatin to affect their immediate neighbors and control the behavior of those cells. So we think that feedback from delta cells, we've shown it's triggered by urocortin-3, and it determines the set point for plasma glucose during postnatal maturation. And it timely attenuates insulin release to prevent insulin secretion from overshooting, which then would need to be compensated by a compensatory release glucagon. So this is an example where local feedback between two neighboring cells is important to determine a physiological parameter. I've also shown you that this intra-islet delta cell dependent local feedback breaks down early in diabetes. It may, on the one hand, be a compensatory mechanism to allow maximum insulin secretion in the face of insulin resistance, but also it contributes to its pathophysiology because it helps explain how tight control over glucose breaks down in diabetes. And then I think delta cells are not only important to explain the actions of urocortin-3. I think delta cells really serve as this local control hub that integrates signals from a variety of different sources, not only urocortin-3, but for example also ghrelin, and can translate this into tonic inhibition on insulin secretion from beta cells. Okay. So the second half of the story I was going to share today has to do with the property that urocortin-3 is this really wonderful maturity marker in beta cells. This particular picture shows postnatal day 2, where a good number of beta cells already expresses urocortin-3, but quite a few of them, indicated by arrows, do not. We've for a while thought that by the time that maturation rolls around, all beta cells express urocortin-3. Well, it turns out that that's not entirely the case. And that's the story that I'll share in the next couple of slides. So a little bit of background. It's been long thought that maintenance and regeneration of beta cell mass is controlled exclusively by slow beta cell self-renewal, and that all beta cells contribute to this process. And this is still the main mechanism by which beta cell mass can be restored or expanded, at least in mouse models. But the problem is that human beta cell proliferation, at best, is a slow process. And the disease where this is most relevant, type 1 diabetes, by the time that somebody presents at their doctor, they're usually already at a point in their disease where they've lost most of their beta cells through ongoing autoimmune assault. So, and then patients are placed on insulin treatment. But to come back from a situation where you have few, if any, beta cells left that are still ongoing or experiencing autoimmune attack, and knowing that beta cells at best proliferate slowly, this is not a particularly appealing target to try and restore beta cell mass in patients with type 1 diabetes. So then a couple of really interesting studies came around that I think captivated the attention of the field by Pedro Herrera, who showed that if you completely ablate beta cells, you can cause or activate considerable plasticity in islet cell fate. They showed in a couple of papers that both alpha and delta cells, under the right circumstances, can transdifferentiate into beta cells. So the story I will show is that we think that there is actually a presence of a population of beta cells that we call virgin beta cells that arises via transdifferentiation from non-beta cell progenitors at a spatially defined niche that occurs at the edge of islets, of healthy pancreatic islets. So we think that this plasticity is not just limited to models of beta cell loss and pancreatic injury, but we think this is a normal part that happens, that a normal part of maintenance of beta cell mass and islet cell mass. And what led us to this conclusion is the following set of observations. This is an image of a mouse islet, where I think you'll agree with me that most beta cells look equal. They all equally stay in the intensity for insulin. And then you see this nice peripheral rim of alpha cells labeled for glucagon. Now, if we start to overlay urocortin-3 labeling, you start to see significant heterogeneity. Not all beta cells are equally green. And if you look carefully, there's actually a couple of beta cells that are clearly red. But if you toggle off the red signal, there is no green underlying that red. So these beta cells are not expressing urocortin-3 at all. So the one thing that we also observed about these is that these cells tend to occur at the edge of the islet. Now we wanted to quantify that. So we designed an algorithm that really basically defines both the outline and the center of mass for every islet, and automatically detects the location of each nucleus in these islets. And we can then use this to map the position of every cell in this islet automatically across hundreds of islets and thousands of cells. And the way we do this is we assign a value to every dot, and a value, a nucleus that is close to the center of the islet will be assigned a low value. So we draw a line from the center of the islet through that cell to the edge, to the nearest edge. And when a cell is close to the edge, we will assign it a high value, close to 1. When an islet cell is close to the center, it'll be assigned a value closer to 0. And we can then use this to plot the distribution, cumulative distribution of every cell in the islet, and that curve will look like this for the simple fact that there is more space, more real estate at the periphery of the islet for cells to sit there, and there's very few cells that sit in the very center of the islet for obvious reasons. And if we then start to break down all these cells into their cell types, we can see that mature beta cells tend to distribute slightly more central than the average cell, and alpha cells tend to distribute, in mice at least, very peripheral, which is a recapitulation of the known architecture of mouse islets. So this, in a way, validates our approach. And now we did the same thing for these immature cells. And we've done this over, I think there were 23 animals in this particular study, hundreds of immature beta cells. And very clearly, as you do this, these immature beta cells, as a population, tend to distribute quite peripherally, just internal from the layer of alpha cells, but markedly more peripheral than the average beta cell. And as you quantify this, what we are left with is, in early postnatal development, a large proportion of beta cells that is urocortin-3 negative, but these cells persist, and they persist at about 1 to 1.5% of all beta cells. So I've shown you previously that in diabetes, beta cells lose urocortin-3. So the question then became, well, are these beta cells that have dedifferentiated? Are they diseased or dysfunctional beta cells? Or are these beta cells that have never before expressed urocortin-3 and therefore immature? So we've done lineage tracing, which you can do in mice, to address this question. And when we use a urocortin-3 Cree line to drive the expression of, in this case, membrane GFP as a lineage marker, we can see peripheral beta cells, which are labeled for insulin, which are red. This is the NTMG mouse. So a lineage negative cell will be red, a lineage positive cell will be green. And we can see that these beta cells at the edge are lineage negative for urocortin-3, which indicates that they've never before been a mature beta cell. So this is how we know that these cells have arrived from a non-beta progenitor that has never before been a mature beta cell. So we also have a wonderful bitransgenic mouse line that we developed at our disposal to really isolate these cells and visualize them. And this is a combination of a nuclear cherry driven by the insulin 1 promoter, which labels all beta cells in red, that we cross to a urocortin-3 GFP reporter, which labels all mature beta cells in green. And as you visualize these islets in 3D, you can find these pockets of immature cells at the islet periphery. And as you open up the 3D the 3D islets, the Z-stacks, you can see these pockets of immature cells at the periphery, but the entire core of the islet is uniformly labeled for urocortin-3 and cherry. So these are all your mature beta cells. So using this particular biotransgenic mice, we can then do a FACS-based purification, where we can isolate the M-cherry single positive immature beta cells and the GFP and cherry co-positive mature beta cells, and get their transcriptomes. And when you do this, you find that the immature beta cells, even though they're different from the matures, they still cluster together and separate from all the non-beta endocrine cells in these islets. So these immature beta cells are more beta than anything else, but they're also distinct from mature beta cells. And for example, they have reduced expression of urocortin-3, which is the marker that got us started on this path. They also have reduced expression of MAF-A and increased expression of MAF-B. And these two transcription factors are known to be markers for mature beta cells in the case of MAF-A, and they're known to be expressed in immature cells in the case of MAF-B. So this tracks exactly with what you would expect for immature beta cells. When you do pathway analysis, you can find this is a schematic representation of the beta cell. You can see that basically every single pathway that we know beta cells need to engage in glucose sensing leading to insulin secretion are downregulated in immature beta cells. These are genes involved in the TCA cycle, genes involved in oxidative phosphorylation. They are almost always significantly downregulated in the immature beta cells compared to the mature beta cells. And we can validate some of these also by immunofluorescence. For example, there is an immature beta cell here at the periphery, which is very low in expression for G6PC2, which is required for setting the glucose threshold for these beta cells. And this is also a cell that has no urocortin-3. We've also validated this for ERO1 like B and a number of different markers. So the transcriptional changes that we show hold up very well as we do validation by antibody-based markers. Not everything is down in these beta cells. There's a group of cells, a group of genes that is known as disallowed genes. This is Guy Rutter and Franz Schuit and Anil Bhushan who've forwarded these concepts. These are genes that are known to be expressed by immature beta cells or beta cells during development that need to be repressed as beta cells mature into a functional beta cell. So you would expect these genes to be high or enriched in immature beta cells, and again, that's exactly what we see. And then finally, we've done some of that to physiology, where we depolarized these cells and looked at calcium currents. And whereas mature beta cells respond to a forced depolarization with an influx of calcium, which is what they need to engage in insulin secretion, immature beta cells appear to be incapable of mounting a calcium influx. So they would be functionally incapable of responding, even if they were able to sense glucose. But we think they cannot. So this is another example where we validated our mRNA expression by RNA Seek with antibody-based detection of cell surface GLUT2. And these peripheral insulin positive urocortin-3 negative beta cells, which are highlighted by these arrows, they have no cell surface GLUT2. So they're red, but they're not green, and they also lack their cell surface GLUT2, which is white. So if these cells have no GLUT2 and cannot sense glucose, well, can they take up glucose? So we've actually set this up. What I'm going to show you is a video where I'm going to apply a glucose trace in green. And what you can see over time is that as soon as we apply that glucose trace, it progressively permeates the entire islet and stains individual cells really throughout the beta cell mass. And again, beta cells in this case are indicated by a red nucleus, with the exception of a handful of peripheral cells, which are red. They have a red nucleus, they're beta cells, but they really do not take up any glucose. And that's summarized here, where if you quantify if you quantify glucose uptake, you see a significantly repressed glucose uptake in immature beta cells compared to mature beta cells. Again, as we look at human islets, we can also find evidence for these cells. Now, we have no ability to lineage trace, but what we have done is we've looked at really young neonate human donors, where we can find clusters of cells that are insulin positive, but urocortin-3 negative. And interestingly, these tend to occur at the interface of areas with alpha cells and areas with beta cells. So again, suggesting that there is something special to the area within the islet where different cell populations are. So if these cells are coming from a non-beta cell source, what is that source? And since these cells occur in close proximity to alpha cells, the obvious question, also given the fact that Pedro Herrera showed that alpha cell transdifferentiation can take place, do these cells arrive from alpha cells? So we've done the experiment where we've lineage labeled all alpha cells, in this case in green, and then we've labeled with a combination of glucagon for alpha cells and urocortin-3 for mature beta cells. And we can find cells such as the pair of cells in this particular detail, where we have a pair of cells that clearly is green, so it used to be alpha cells, but they've lost the expression of glucagon, and instead they now express urocortin-3. So they've gone from a alpha cell fate to a beta cell fate. So we used a glucagon cream that is not inducible. So the concern obviously is that this could either be a stochastic artifact of the line, where we just randomly label a low frequency of beta cells. If that were to be the case, we would expect to find random beta cells across the surface of the islet to be labeled for lineage labeled for glucagon. It could also be that during embryonic development, there is a stage where a progenitor cell is transiently co-positive for insulin and glucagon before it goes on, becomes a beta cell, but in the process picks up an alpha cell lineage label. If that were the case, as islets are formed by expansion of the beta cell mass, one would expect to find clusters of labeled beta cells at random locations in islets, and we do not observe this at all. Instead, what we observe is that we see these lineage labeled beta cells. The beta cells that have an alpha cell lineage label occur only at the periphery of the islets, and we can quantify this by using the exact same distribution trick that we developed earlier. And if we do this, we can very clearly see that these beta cells with an alpha cell lineage label occur highly selectively at the edge of the islet, again, just internal from the alpha cell mantle. And just to show you how this fits in with these immature beta cells that I started with, we think that these immature beta cells that express insulin but not yet urocortin-3 are a transitionary stage that is on its way to becoming a beta cell. And that is a couple of examples here. In this case, two different cells that both are lineage positive for alpha cells. In this case, they're green. The bottom cell already is positive for both insulin and urocortin-3, so it's completed its transition from an alpha to a beta cell. But the top cell is positive for insulin, but negative for urocortin-3. So this is an example of an immature beta cell that is coming from an alpha cell fate. So it's en route, if you will, from alpha to beta cells. It has lost glucagon. It has acquired insulin, but it has not yet acquired urocortin. All right, so we can also isolate these cells. This is a strategy that requires 3 different transgenes. Again, our mouse insulin and cherry reporter labeling all beta cells in red, and then a combination of glucagon Cree and YFP to lineage label all beta cells. And you can see these clusters of cells at the periphery that clearly have a red nucleus but are green. So they cannot lose the YFP, but as they become beta cells, they acquire a red nucleus. So this allows us to fax purify these cells. They're rare, but they exist, and we can get enough of them to get transcriptomes. And as we look at the transcriptomes of these transdifferentiated cells, they are more similar to beta cells than they are to the alpha cells that they once were. So by all indications, these cells look like beta cells. They've lost glucagon, they've acquired insulin and urocortin-3. But the big question was, do these beta cells of alpha cell origin actually behave like a normal beta cell? So they look like beta cells, they have all the right markers, but what is their functional behavior? So we tested this, and we did this by using a calcium indicator called GCAM6. So we expressed GCAM6 in a Cree-dependent manner in alpha cells, which, like before, is a lineage label, but at the same time allowed us to dynamically to assess the behavior of these cells in intact islets over time. So we're combining these 2 properties of this particular flox allele. So what I'm going to show you here is, before we go into the transdifferentiation part, is a movie where I'm going to indicate or illustrate the prototypical behavior of alpha cells and beta cells. So on the left, you're going to see an islet where all beta cells are labeled in red, and all alpha cells are lineage labeled for GCAM6. So as the time course starts, we'll apply AVP, which is a peptide that is known to activate alpha cells. And every time we apply a puff of AVP, we see alpha cells twinkle individually. There's no coordination amongst them. They each respond to their own rhythm. And then there's depolarization at the end, just to show that they are viable throughout the whole experiment. Now I'm going to show you an experiment with beta cells. Again, all beta cells are labeled in red, but now we also express urocortin GCAM in beta cells. And instead of AVP, we apply glucose. And we see this very nice pulsatile behavior to continuous glucose, which is well known and a hallmark of healthy beta cell function. But notice how this is much different from the response of alpha cells. So now that we had established how beta cells and alpha cells ought to respond normally, again, alpha cells respond to AVP in a non-coordinated fashion. They each respond individually, whereas beta cells have this very nice pulsatile pattern that is mediated by gap junction formation. We can now start to look for evidence of functional maturation, or functional transdifferentiation. Again, we use an insulin M cherry labeling all beta cells in red, and we lineage label the entire alpha cell lineage using GCAM6. So when I play this particular movie, you're going to see a close-up of a single islet, where now I'm going to apply first AVP and then glucose. So as we apply AVP, you see individual alpha cells twinkle, just as before. So these are your conventional alpha cells that respond to AVP. Now we're switching to glucose, and now you see this different group of cells that is pulsing to glucose, just like beta cells would. They're at the periphery of the islet, and they have a red nucleus. So meaning that they are currently beta cells because the expression of M cherry is contingent on insulin promoter activity. So as we summarize this particular video, there is two clusters of cells. There is one cluster of cells that responds in typical alpha cell fashion to AVP and then twinkles, and then it doesn't really respond to glucose, and at the end responds to depolarization. And contrast this to the second cluster of cells, which has no response to AVP whatsoever. They've lost all alpha cell traits, but they start to break out in this beautiful pulsatile secretion in response to glucose. So these are cells that then have completed their transdifferentiation from alpha into functionally mature beta cells. So the final piece of data I'll share is that this is not limited to alpha to beta cell transdifferentiation. We think this happens both ways. This is a particular example where we've lineage labeled all beta cells. So all beta cells, all mature beta cells are green. And we see examples. This is an example of one image where there's two cells that no longer express urocortin 3. So these are cells that once were mature beta cells, have lost urocortin 3, and now acquire glucagon. And we can actually, this is the final video I'll show you, we can actually also visualize the behavior of these cells. So these are two different islets, one of which where alpha cells are lineage labeled with GCAM6, and the second islet, all mature beta cells are lineage labeled GCAM6. And we're going to take this pair of islets through the same stimulation protocol as before. So again, it's two islets, glucagon lineage labeled on the left, mature beta cell urocortin 3 lineage labeled on the right, and we're going to first apply AVP. And there's these two cells on the beta cell islet that responded to AVP. They do not respond to glucose, in contrast to all the other beta cells in the same islet. And then at the very end of it, they come back and they respond to KCL. So again, there's these two cells here that as soon as we apply AVP, these two cells that are lineage labeled by urocortin 3, they used to be mature beta cells. They respond to AVP, just like the alpha cells in the neighboring islet. They do not respond to glucose, unlike all the beta cells in this particular islet, and they all respond to KCL-based depolarization. So this is an example where, again, at the periphery of the mouse islet, mature beta cells transdifferentiate into alpha cells. So this is not a one-way street. We think there is this constant exchange in cell fate taking place at the periphery of the islet. So to summarize this, it's still true that cell replication is the main mechanism of beta cell mass maintenance, at least in mice. But this is not a particularly appealing target in cases of extreme beta cell loss, such as human type 1 diabetes. So what we've discovered is that transdifferentiation between alpha and beta cells, both ways, takes place at a neogenic niche, a specific location at the periphery of healthy adult islets. And the urocortin-3 negative immature beta cells that we discovered, we think are an intermediate stage in those transdifferentiation processes. And we think that this transdifferentiation is driven by the complex combination of signals that converges locally at the niche. These signals may have nothing to do with pancreas injury or repair. They may be part of the normal complex mixture of signals that could be neurotransmitters, that could be alpha or beta or delta cell derived, that can be found and that come together at the edge of the islet. And we think this is exciting simply because these cells exist, but I think it's also exciting that we find this ongoing maturation of beta cells in healthy islets, because this exact maturation is what's the current bottleneck in generating beta cells from stem cells. So by studying beta cell maturation at the periphery of mouse islets, we can actually learn what distinguishes a functionally mature beta cell from an immature beta cell. And this will allow us to learn how we can turn immature beta cells or beta cell-like cells and make them into true functionally mature beta cells that are capable of meaningfully restoring insulin production in people with type 1 diabetes. So with that, I'd like to thank the folks in my group who've done a ton of this work, particularly Talita and Alex and Michael, who contributed to the last story that I shared, and then Talita along with Cindy and Joan, Vera, and Sophie, former members in my group, who drove the story on the crosstalk. A wonderful set of collaborators. Michael was responsible for some of the algorithms that we generated, long-standing collaboration with Maike. Klaus and Amanda were generous in supplying the glucagon-free ER mice that I didn't show, but were instrumental in us making the case that we see transdifferentiation in adult islets. And then human pancreas slides from the NPOD consortium, live human islets from the IIDP consortium, and funding from the Juvenile Diabetes Research Foundation and the Hartwell Foundation. And with that, I thank you for your attention.
Thanks, Mark. It's really nice. You gave a wonderful overview of how the things work with the urocortin and the differentiation between alpha, beta, the cells, how they. I have a couple of questions because we are almost out of time regarding your first part. You can hear me, right?
I can hear you just fine, yes.
Is there any specific receptor for urocortin on the delta cells, like where they helps in the secretion of some other strategy?
Sure. So the receptor for urocortin-3 is the CRH receptor 2. It's a class B G protein coupled receptor. We know that, as far as we know, and I think we have a good sense, this is the only receptor that is capable of responding to urocortin-3, and it is selectively expressed by only delta cells.
Okay. And the other question in the same thing is, like you have mentioned that, and you have shown the data that urocortin goes down during diabetes and all. Can it be used as a pre-diabetic marker? Like, hey, this person might be getting that. Now it is basically the triglycerides and all those things. It doesn't, I mean, like, for example, my triglycerides has been up for last 10 years and I'm not diabetic. So it doesn't mean anything. So the question is, can this be used as a marker? Hey, this is really like in a couple of years, you might get diabetes for sure, kind of thing.
I think particularly the macaque data, which are just a wonderful opportunity for us because there's so much more control than whatever samples we can get from NPOD, which just has more variation to them. I think the macaque data argue very strongly that this is something, this is a protein that is downregulated before frank diabetes. The missing piece, the thing we're going to have to solve is how are we able to measure it systemically. I mean, we can measure it clearly on biopsy samples, but that's not an appropriate way to diagnose prediabetes.
Yeah.
So
it relates in the blood?
I mean, it's detectable in blood, but as I indicated, I think all of these signals are really local signals that are released in high enough quantities to reach the neighboring cells, either through the capillaries or through the interstitial compartment. There's not enough of them there to be released into the systemic circulation. So measuring them in systemic circulation, the sensitivity of that measurement is going to be the bottleneck in whether we're able to say something or use urocortin-3 as a marker to indicate ongoing dysfunctional beta cells and prediabetes. I think it's potentially possible. I think that's where we're going to have to make some improvements if we wanted to go down that path.
Yeah, I mean, like that would be, I think that is one of the real, like, with your data and other data, it shows that it goes down.
Mm-hmm.
Hey, there is something wrong with the beta cells. And like we have glucose tolerance test, right? Like something like that, there is some way you can measure it and show that, hey, certainly going to get diabetes in few years.
Mm-hmm.
That might be a real way to change the lifestyle or whatever you want to do for the treatment. Yeah, it's a very interesting. I'm really appreciate you coming on and show us your work, and I think everybody would like your work, and we'll put it out on our website immediately. You'll see it, and on our homepage in the next couple of days.
Okay.
And again, thank you so much, and I appreciate your time.
I thank you very much for having me on. I appreciate it.